combining some lab work back in Richland with some work right here at Hot Lake and in Oroville as we're doing now. So first off, Pacific Northwest National Laboratory, PNNL, is one of a number of Department of Energy labs across the country. So, you know, we're obviously up here down in southeastern Washington. Maybe not as widely known as some of the other national labs, but I think, you know, you've probably all heard of Los Alamos, maybe Sandia, Brookhaven, Lawrence Berkeley, Lawrence Livermore Labs, Oak Ridge, some of those. But we're right out there. We are an Office of Science lab. So most of the buildings that we work in are owned by DOE, and you know my computer has a little DOE sticker on it, so it's owned by the Department of Energy. When you look at the lab itself, we have used to be 4,300 folks working there. We're closer to 4,000 right now. Due to some budget.
At your location?
What's that?
At your location?
At our location, yeah, in Richland. And this here's some numbers. You know, we have a lot of peer-reviewed publications, mostly in science, some in engineering, even computer science, things like that as well. A lot of patents. These are kind of per year here, and some awards as well. Now, we get funding from a lot of different places. Department of Energy provides most of the funding that we have, and this is split into different areas. One is national security, then there's energy and environment, and then science. And the project that we're working on is funded by the Office of Science within DOE, so it's in this moderate to smaller slice of the pie. But we're very, very thankful for their support. And I should say, I don't know, I have not been to the border crossing up here, but one of the things that PNNL has done is helped install these radio portal monitoring projects. I believe at every single border now, if you were to drive through there with a lot of radiation, it would set off an alarm. Someone would know, right? So that's one of the more recent projects that PNNL has done. And the other sort of national security thing that they've done that I should mention, I don't know if anyone's flown on an airplane recently, but you know they're moving away from the X-ray detectors a lot, and they're doing you get a little round thing, you put your arms up, and they scan you. And PNNL was very instrumental in developing one of those two types of systems that's being used in airports right now. And they're still doing some research on trying to fine-tune those. So the goal of PNNL, this is the branded goal, is to transform the world through courageous discovery and innovation. And that's mainly done through, as I said, engineering and scientific work. Almost everyone that works at PNNL is on the Richland campus, although there are some very small offices in Seattle. Maybe a couple hundred people, 100 to a couple hundred people over at SWIM. They do primarily estuary research over there. In a very, very small office in Portland, and just a couple people really over in DC that work for PNNL. So this is the majority of our main campus. There's a couple other buildings. This is down in Richland. A few interesting things to note is that this part of campus is all nice and green, and there's some trees around it and all that. And unfortunately, my office is over there. So I don't have the nice green trees. They stuck me out in the desert, but that's okay.
Riverview, though.
Riverview, yeah, and it's really nice, especially in the winter. You know, the bald eagles and stuff come down from the mountains and hang out in the river.
I thought you're acclimated to the field.
Yeah, it's nice that way. Right? We don't want those.
Right.
I'll help you there.
Thank you. I wasn't expecting that.
The other thing to mention is that there's an invisible line right about here, and north of that line is the Hanford site. So we were talking a little bit before about the Manhattan Project. I don't know how much you know, but certainly north of that line was very instrumental in a lot of the Manhattan Project activities. And there's still some interesting sites to visit up there if you ever get a chance. So as I mentioned, our funding comes primarily through the Office of Science from the Department of Energy. And our specific project is this. It sounds like a mouthful, but we're studying the principles of microbial community design. So what makes the microbial community develop in the way that it does? And we'll talk about that a little bit more. First, another jargon-filled sentence perhaps, but this comes from DOE. They are interested in understanding the fundamental design principles of microbial communities and how such principles demonstrate predictive behavior of complex, heterogeneous systems and to the design and control of stable communities in engineered environments. It sounds like a mouthful, but I'm hoping it will convert that to English. You notice that's even just part of one sentence. These people, they just think in like these long, crazy sentences. But what we're really trying to do is look at the driving forces and mechanisms involved in microbial communities. So what forces them? And you know, microbes are a lot different from people, obviously, but it's really the same thing as like food, right? Food is a driving force for us. It's really a majority of some of our work now at Hot Lake is what is the food for these different microbes. And then principles controlling spatial and temporal dynamics. That is just, you know, fancy jargon, like why do these things organize in the way that they do? And we'll see some pictures very soon of some of the mats that we're studying. And they're very highly organized. There's different colors and different layers. It's like, well, why do they do that? And you know, how do they organize themselves along those spatial scales? And then, you know, when we're going to look at this in much more detail, but there's a lot of different organisms in microbial mat. Why do they need each other? So how do they interact with each other to make one community instead of just living on their own? And we'll look at some more slides from that, but please stop me and ask any questions if you have. You know, Clyde said I had to be less than an hour, and I don't think that's going to be a problem. But so feel free to stop me if I gloss over things too quickly, and sometimes, unfortunately, I do. But the previous slide is some of the things we're looking at. The first question that a lot of people have is, why? Who cares, right? And so there's a lot of reasons why you might care. This is just one example. This is an example very relevant to the Department of Energy. It's also an example of something that our research might lead to in the future, well in the future. So we're not going to be dealing with this in the next year or two. But this is many, many years down the road. And we're hoping to lay some of the foundation for it now. But what you have here is an artist's picture of a bunch of ponds, and they're all growing, say, algae or something like that. Put whatever algae microbe they want in there. It grows really well. It looks green and beautiful. It'll be going fine for a while, and then it just dies, up and dies. And the whole population just crashes. And they're trying to figure out, well, why? And one of these things that you know about the natural world is that the more different species are interacting in a microbial community, the more stable it is. So when you just put one algae in there, okay, it'll be fine, and then it'll just die. And they don't really know why, though. You have more species interacting, why it stabilizes the system. So that's one of the things we're looking at. And again, it's just a small piece of it. It's kind of a grand challenge, but why do microbes interact the way that they do? And why does that make the overall community stronger, more resistant to change, more active, more able to maybe make more biofuels? So we have two main field sites for this project. One of them is in Yellowstone National Park, and this is boiling water, right? So it's really, really hot. Unfortunately, it boils sporadically, so if you're standing too close, it'll spout a little bit, and I've taken a few mouthfuls of boiling water, and it's not much fun. This water is very close to boiling. This water is extremely acidic. This water is not acidic. They're both very, very hot. This water has a lot of dissolved iron. It's the oxygen, and the reason this is all orange is it's iron oxide, which is basically just rust, right?
Where is that one on the left? Location.
This is in Norris Geyser Basin in Yellowstone, and this is in Lower Geyser, pretty close to Lower Geyser Basin.
Okay, so
This is Conch Spring, and this is 100 Springs Plain Spring. So, and the conditions here are so extreme that the microbial communities are not as robust, or not as diverse, maybe, as you might find in a lot of areas. So if you go out into the soil here, there might be hundreds, thousands of different microbes interacting. Well, here, life is really rough, so there's a fewer number interacting, which makes it easier to study. Because if you're trying to study interactions between thousands and thousands of things, it's harder than if you're trying to study interactions between 20.
Is that statement based on that site, or are you talking Yellowstone in itself?
Yellowstone, the vast majority of sites in Yellowstone. These systems are so different that different organisms live there.
Yeah.
That's very acidic. This is not acidic. It's pretty neutral.
But they're still small.
There's still fewer things able to live there. I mean, if you wanted to study microbial ecology, if you just went into a healthy farm pasture, you could probably spend almost your whole life trying to figure out what organisms are there. It's just so complex. So
There's a few people that have done that.
that have done that? Okay. It's just so hard. Now, the other site that we have is the Hot Lake system. And this is some of the microbial mat that grows at Hot Lake. This is from last year. Looks a little bit different up there right now. It changes seasonally, changes due to a lot of different conditions. But what we have up at Hot Lake is also a very extreme environment. It might not look extreme here, but I'll show you a couple slides on it. The salinity at Hot Lake is extremely, extremely high. And that salinity has had some very nice implications in the past for economic development. The salinity is largely, all that salt in there is largely basically Epsom salts. And back in the World War I era, folks went up to Hot Lake and they were actually mining it. And here's an old picture of it. But apparently the Epsomite that came out of here, Epsom salts, was amongst the purest Epsom salts ever mined in the U.S. Instead of, you read some of these old accounts, and basically they built a system to purify it, but they're like, well, this is just for show because it's already so pure, we can just sell it to the people that need it. And that didn't last that long. I don't completely understand the motivation behind this, but I have been told that Epsom salts can be a component of gunpowder, and that prior to this, we got a lot of gunpowder from Germany, and for some reason in World War I, Germany stopped selling us their gunpowder ingredients, so we had to go here and find our own. After the mining, things were pretty quiet up there, and then there was a period. These are a couple pictures taken from the 50s, where there was kind of a resort or spa out there to take advantage of potentially healing waters that people would soak in out there. Now,
That's Dorothy.
Dorothy, yep. She provided you provide us these pictures and others.
Yes.
Yep. And after that, currently, I don't know how long ago this was, but as right now, Hot Lake is owned by the Bureau of Land Management, by BLM, and they've set it aside for research. So it's an RNA, or a research natural area. And I think we're really the primary people that are using it right now, but it is open. Anyone that wants to do research up there, they can apply and get a permit.
Is Epsom salts still being reclaimed or not?
No, there's no mining up there now.
I'm in a natural state.
Is there still Epsomite up there?
Yeah.
Oh yeah. Yep. Yeah, definitely.
Those two pictures. Two different.
We're not the first folks to do research up there.
Yeah.
Jenkins, back in 1918, did some great work on the geochemistry of the system and really helped define why there's so much Epsom salts in there. This is a picture, I have not been there, perhaps many of you have, but of Spotted Lake, just north of here in BC. And I think it looks, you're shaking your head saying you've seen it, and it looks like this. There's a bunch of...
Yeah.
Spotted Lake. Okay. So yeah, don't hide the view of the lake though with the billboard, okay? But apparently there's a lot of round little pools, and we think that's what Hot Lake looked like probably before the mining. But then after the mining, they sort of opened up these larger basins. So instead of getting a lot of these little things, you have slightly larger basins up there. So Jenkins back in 1918 did some great work, and then Anderson out of UW in Seattle, he did some work in the late 50s. And he was looking at a little bit of geochemistry. He did a little mapping of the lake, and then he was looking at some of the life in there too. So some of the brine shrimp that live there, some of the plant species around the lake, and things like that. Now, an interesting thing, his paper, a couple interesting things about it. First of all, this was the most recent paper published on Hot Lake until very recently. And secondly, this being the Chamber of Commerce, in Anderson's paper, he got a lot of help from you guys. And he cited you in the paper. So back in 1958, the Oroville Chamber of Commerce was cited very well. I don't know. Yeah.
Since we've only existed legally since 1972, who knows?
Yeah, well, he gets in print, so I don't know.
No matter what, senior chicken farmers, they do underground.
Yeah, the underground Chamber of Commerce. So this is an aerial view of Hot Lake. I'm not sure if everyone would call it a lake. Its water surface area varies tremendously based on the weather. It's probably something on the order of 3 acres, maybe water surface area, but again, it's highly variable. What you can't quite make out from this picture, you can trust me for it, which I don't know if you want to do that or not, but there's a ridge that goes all the way around here. This lake is landlocked. There's no river or stream that goes into it, and there's no river or stream that goes out of it. So any rain that falls here and washes into the lake, it's there until it evaporates. And Anderson and some others have looked for groundwater inputs to the lake. There's no groundwater inputs that we know. So it's like, as far as we know, the water in here comes from precipitation almost directly, and it sits there until it evaporates. Well, anytime you wash a whole bunch of water into a lake over time and then evaporate the water out, the residual water gets a little bit salty. And that cycle's been happening over and over, washing in, dissolving a little bit of the rocks, washing it into the lake, and then the salinity builds up and most of the salts won't evaporate. The water evaporates and leaves the salts behind. And that's largely how the lake got so salty. And if you look at it, I mentioned that right now, we have our weather expert in the room, who knows more than I do, but the rain around this area is not consistent, right? You get much more rain and much more precipitation in the winter. Well, in the spring, when you have your snow melt and rain events, the lake level tends to increase a bit with more water in the lake. And then over the course of the summer, when it gets hot and dry, especially this summer, it evaporates quite a bit. The lake level goes down. As the lake level goes down, you're taking all that salt and smushing it into less water. The salinity goes up quite a bit. So these are old numbers, but in April of 2011, the salinity in the total dissolved solids in Hot Lake water was, let's say, 35, 40 grams per liter. So first of all, that's a huge number, right? When you think about it, if you have a liter of water, which is about a quart of water, right, and you evaporate it away, you get 35 grams of salt in the body. But that was in April. Once you start evaporating the water in July, September, and October, you notice that the salinity just goes up and up as you're evaporating more and more of that water. And the salinity stays up until you start getting fall or early winter rains, and then it brings the salinity back down. The interesting thing to put that into perspective is that this spring was pretty close to the same salinity as ocean water. Ocean water, pretty salty, right? But not nearly as salty as the Great Salt Lake or the Dead Sea. And by the time you get to the late summer, the Hot Lake salinity is up to about the same level of the North Arm of the Great Salt Lake and approaching that of the Dead Sea. So it's kind of a cyclic pattern, but you get a lot of salinity up there. And the microbes, this is the cool thing for me, okay, there are microbes starting to develop in April and May. These mats are starting to grow there. And they're there throughout the summer. So they have to change, or withstand a very large change in salinity. And, just with, we don't have a very large capacity for salinity. If we drink salt water, try that for a couple of days and I think you'll be in trouble. But these mats can handle it. And it's just remarkable. Not only handle it, but handle the huge change as well. The other really weird thing about the lake, up at Hot Lake, is what makes up the salinity. So salinity is made up of all the different salts. And we're just naturally, I think, used to marine systems. And marine systems are largely sodium chloride. These guys, which is the same thing that's around the table right now, so it tastes good, right? But Hot Lake, the salts are dominated by magnesium and sulfate, which is very rare. There aren't many lakes in the world that are dominated. There's a few in this region. There's also a few in the Geochemistry region in Spain. But outside of that, almost all hypersaline environments that have been described in the literature really basically come from marine salts that have dried up. And so really they're dominated by this sodium chloride. So Hot Lake is very unique in that it's got tons and tons of salt, so the microbes have to withstand that. So we want to know how they interact to withstand that. The salts are different than those in almost any other microbial environment around the world. So it's kind of a unique system in that way. Plus, the salinity goes up and down over the course of the year, so they have to withstand a lot of changes. That all makes it a very interesting place to study some of that microbial life. Now, we are very interested in understanding the effect of the rain and evaporation on the salinity levels. So myself and a couple of others of us went up in November in 2012. This is an aerial image of Hot Lake, and every one of those red spots right there is a survey point. And we went and we stood out there. It was kind of cool for part of that. We tried to survey the lake to really get a good feel for what the shape looked like. And we developed this map, very similar to a topographic map. And I'll show you better in the next slide. But you can see these large basins. There's not many topo lines on them. They're pretty flat on the bottom. And again, we think that's probably a remnant from some of the mining that went on up there for the epsilon. And if you look here, hopefully this will play, this is kind of a color image of the map that we made. You can see some of the major basins. And then in a second, it's going to turn, and you'll also see right here how they're nice and flat, sort of regions on the bottom. And in the side, it's a little bit bowl-shaped. And then once you get above that bowl, there's sort of a bowl and then a big fan on top, a big plane on top. And that's what right now, the water level is sort of really low, so it's down in these bowls, kind of where the steep sides. So the lake looks really small. Earlier this spring, it had filled the bowl and then was out on the plane on top, so it looked a lot bigger. But there wasn't necessarily a lot more volume there. It's just a lot more surface area.
So, Jim, you think that you think these basins were caused by the mining of the Epsom salt?
That's what we think. But anyone here might know differently.
Well, and if that's the case, I mean, if you dug down below the level, the shallow basin area, would you find, or have you found collections of Epsom salt?
Oh yeah.
So that it goes down to maybe what the original level of the lake was hundreds, thousands of years ago?
Potentially. Well, yeah, probably. We don't know, but it's probably a fairly young lake. Okay. Probably from the last glacial age, or something like that. So 10,000 years, something, or whatever. Yeah, fairly young, right?
But they did mine underneath it. There was a mine under it, and so it's a cave underneath it. So if they do go down, they're going to bust through that.
Yeah. Would have gone, yeah. But Epsomite right now, you can take a pole and go along, say you're in a little raft and you go along here, you can tap the pole on the ground and it's like, oh, mud, sort of muck, that stuff you don't want to step in. But then once you get to one of these deeper spots, it's just clunk, clunk, clunk. And if you, on a long stick, my arm's not long enough to reach down there, but if you scoop some of that up and bring up, it's all little crystals that's largely Epsomite.
So it hasn't, the muck hasn't settled in these big spaces?
No, the muck hasn't really settled in. There's potentially a couple reasons for that, and one reason, I have a slide on it, so I'll save it to that. I'm trying to remember what I have here. So this is, we're thankful for some of the weather data that comes out of this area, right? But this is from last summer. And you see here, we in the purple here, we have precipitation. This is, I failed to label the graph, I'm sorry, but this is Julian day, so this is, 1 is January 1st. 365 is December 31st. So you can see just about how much precipitation comes over the course of the year, and you can see where summer and fall is pretty flat. There's not really much added precipitation at all. But what you see is the lake volume goes up early, and then all summer it's sort of going down, and then it cools off and it actually levels out. You also see these crazy words that I'm trying to learn. There's all these limnology words out there, but mixolymian and monomylian. Mouthfuls, right? Complete mouthfuls. But they actually make a little bit of sense. Mixo is it's the region of the lake that mixes, and monomylian is the lake that sort of doesn't mix. And this is a really weird lake because almost every lake you go to, let me see, almost every lake you go to, if you've been swimming, the warm water's on top, you dive down and it gets cooler. Hot Lake's the complete opposite. Hot Lake right now, the top water is maybe lukewarm. It's cold to the touch, but not frigid. The bottom water below is approaching 100 degrees Fahrenheit. Wow. We measured that this afternoon out there. And the reason for that is all the salinity. And this sort of gets back to your question, Clyde. This is the mixolymian. The mixolymian has all about the same salinity. The wind blows, and it mixes it. The monomylian on the bottom, once you start dissolving a ton of salt, the water basically gets heavier. So if you put, the water that we're drinking weighs about 1 gram per milliliter. That's sort of a set in stone. Water at Hot Lake on the bottom of the lake weighs about 1.3 to 1.4 grams per milliliter. So you mix water that's really heavy with water that's lighter, the heavy water goes to the bottom. Then no matter, the lake's fairly small, no matter how hard the wind blows, it doesn't get in, there's not enough ice there to really mix those two layers. So we have one layer on top, which is this layer, where the wind is strong enough to kind of mix things. All the salinity is the same, the temperature is the same, all that's the same. And then below that layer, right below that divide right there, the further down you go in depth, the saltier it gets. And this water weighs more, and there's no way to get enough energy to mix it up. And this part of the water here, I think this is the lake depth. So this top water on top, that's wind mixing and all that. Sunlight goes through that, and due to the microbiology, there's a bunch of different pigments in the bottom water. Sunlight goes through the top water, hits the bottom water, and warms it up. And most water, a cool night, maybe you see fog on top of the water, right? Well, Hot Lake, the warm water, never gets to the surface. So it's almost like there's a blanket. This water that's perched on top that the wind mixes is almost like an inflating blanket on top. So sunlight goes in, gets absorbed in the bottom, and the bottom water never gets to the air to release that energy. So in the summer it gets very hot, like burn you hot for part of this last summer. And it's definitely cooling off a lot now. And there have been reports in the literature that the lake doesn't tend to freeze. We've seen that the lake has frozen when we've been up there. We probably haven't been, but we walked on the ice on it and all that, and it's been pretty well. And I think the reason that it freezes is right now, if you get a lot of rain and then it were to get cold, that really, really fresh water has almost no salt, so it might sit right on the top of the lake. And I think that could potentially freeze.
So have you measured then in the wintertime when it's frozen on top how warm it is down below? So that
yeah, and it's warm, but 50s to 60 maybe, something like that. So not hot, right? But much warmer than you would expect a lake to be.
Yeah. I might have missed it. Why is it warm?
Yeah, it's basically this, and you didn't miss it. I just didn't explain it very well. So you got to help me with that. But there's the water at the bottom never gets to the top.
Okay.
Basically, so the top water is not quite as salty. When you go out there, it tends to be much more clear. So sunlight goes through that top water. The bottom water, I hope I have a picture of it.
So it's solar radiation.
It's all solar radiation. There's no geothermal heat.
Okay.
It's all solar radiation. But the sunlight heats it up, and it never has a chance to hit the atmosphere to release some of that energy. So it takes a long time to get rid of it. So all summer it's just baking and getting warmer. So, okay, great. That's a little background on the lake, but what are we trying to do? Because we're not trying to understand the whole lake. That's a little beyond us. But we're trying to understand really the microbial mat. So in the background here you have a picture of this microbial mat. Last year it was about this thick. This year it's a little bit thinner because there's been a little bit less water.
Is this at the bottom? Where does this exist?
Yep, it's a benthic mat. So it's not floating in the water. It's on the shoreline, the whatever you call the side of the lake. It doesn't go all the way to the bottom of the lake though, because it gets just too hot and too salty for the mat to grow on the very, very bottom. So it's just kind of perched on part of the side.
But it's underwater.
Completely underwater. Yeah. Most of the time it's underwater, except when the water level evaporates all summer. Sometimes when it gets dry and dries up and gets all salty and crusty. And, yeah.
Ooh,
Yeah. So what we're interested in, some of the first things that we're interested in is, let's say we have 40 or 50 different organisms in there. How do they share what they're eating? How do they share vitamins? So, we have to eat, a lot of microbes are like us, they can't make all their vitamins. So they need some microbe that makes them and they got to share them. How do they share their other nutrients? So, they need nitrogen and phosphorus and carbon and all that sort of thing, just like we do. Some of them can make that and some of them can't. So how do the ones that can't make that, how do they get it? And it comes from the other microbes. So how do those interactions happen? And how do some of those interactions improve resistance to change? So let's say the salinity goes crazy. These things are working together. How does it help them to survive? And by working together, do they get overall more biomass? Do they grow more? Do they interact with each other more? And does the whole mat get more stable and prevent the whole thing from just up and dying like a single organism might do? We want to know how these things change during the day and night. Just as an example, during the day, sometimes if you go to Hot Lake on a really sunny, bright day and the mat's really cranking, you can see little bubbles coming up from the mat, and that's oxygen, right? It's doing so much photosynthesis that it's just spitting out oxygen. Well, if you look at that same mat at night, instead of spitting out oxygen, it's just absorbing as much oxygen as it can, and it's got a very sulfitic smell, so the rotten egg smell, which sulfide and oxygen don't coexist. So it goes from one extreme to the other, and during the day there's so much oxygen there, it's just bubbling away, and at night there's almost no oxygen there whatsoever. Let's see, I went the wrong way there. So this is our general experimental plan. So we want to look at environmental dynamics. So certainly for Hot Lake, we've been looking at some of the geochemistry, looking at some of the impacts of weather, solar cycles, and over the course of the season, things like that. And then here, they call it member genomes. It's looking at the DNA sequences. basically, to find out which organisms are there. And as you change these conditions to certain organisms become more or less dominant, did that give you any clue about how they're interacting? And then start probing these interspecies interactions to find out how they're hopefully working with each other. The part of the project that I work on is basically looking at field samples. Here's some nice mat from last July or August at Hot Lake. And then folks have taken that, and basically what they've done is there's a number of photosynthetic green organisms in there. They've pulled apart some of those individual green organisms, mostly cyanobacteria, put them in jars and cultured them. And now what's happened is it's not a pure culture of just that photosynthetic organism, but instead other organisms sort of were attached to that cell or otherwise came across with them. So these simplified natural communities, as we're calling them, contain only organisms that came from Hot Lake, but they don't contain all of the organisms. So they're smaller versions with some of the functionality, but not all of the functionality. And here's two examples. We have Anna over there joining the culture, and Oscar over here. Hard to see because I picked 4 pictures, but they are a little bit different in color. They do behave a little bit differently. And Oscar has this primary photosynthesizing organism. Anna has this guy. We named them Oscar and Anna because we thought that under a microscope, the photosynthetic organism here looked like Annabina, so it became Anna. Oscar looked like Oscillatorius, it became Oscar. Turned out we were just way wrong, so the names don't make any sense, but we stuck with them. So we have the 2 photosynthetic organisms, and then these guys are all things that don't make their own food. They have to get it from someone else. And so these guys are actually shared. They're found in both of these simplified natural communities. This one's only found in Oscar. So we have sort of simplified versions of what's in Hot Lake. And I'm going to give you just 2 brief examples of what we did with it. So this one is Oscar under a microscope with some crazy staining going on there. These long filaments are the phototrope. So these are the green guys. They're cyanobacteria. Oscar itself is a cyanobacteria. It is green. It does photosynthesis. We think it's probably putting all the carbon, all the food into the system. And there's another one here. Then they're quite a bit smaller, but you can see all these sort of cells, mostly rod-shaped, kind of round thing. Those are all the other organisms present in this culture. What we did is we put in a nitrogen source. So this has ammonia, which some of the microbes can actually eat. And we did a little isotope label on that, a stable isotope label, so it doesn't contain anything. And we incubated it here. And then these spots right here that are very, very red or bright yellow, those are the areas that ate more of what we put in. So they took up ammonium quite a bit faster than some of these sort of cooler blue colors. So what we're trying to do here is find out, okay, where does the label go in? And then if we do a time series, like, okay, how does it start spreading? We have maybe single points that the label goes in, or at least there's certain organisms that take it up first, that eat it first. How does it then get to some of the other organisms? So that was one example of the simplified natural communities. And the other is we started looking at some of basically the carbohydrates and sugars that are in this thing. And so here we have Anna, Oscar, and then this is actually true Hot Lake mat from up at the lake. Whatever you want to do to try to interpret this thing, right? This is our typical chromatogram, but just look at the spikes. All the spikes in the chromatogram correlate to different compounds. So, like L is trehalose, which is a two monomer sort of carbohydrate sugar. That thing. Sucrose is, what you put in your coffee. Basically, the sugar we all eat. So you can see that sucrose, Oscar makes it, it's found in the lake, but this Anna simplified natural community doesn't release sucrose. So we're trying to pull apart a little bit which organisms, where in the mat they eat things first, and then which of these simplified natural communities makes things that other organisms might eat. So hopefully that makes a little sense, or let me know if it doesn't.
The retention time, what did that measure?
Yeah. So you're talking about this?
Yeah.
So these are all from gas chromatography. So the way that works is you take a sample, dissolve it in a solvent, usually inject it into the front of a really long glass capillary that's got a stationary base in it, basically. And you heat it up enough to volatilize whatever it is you're looking for. And these things, I don't know how, they take variable amounts of time to get through this gas capillary based on the chemical features of the compound you're looking at. So basically here we have sucrose and trehalose. Sucrose goes through the column faster than trehalose. And this is here we've applied it to gas chromatography, mass spectrometry. There's a lot of liquid chromatography. It's a very popular technique when you just have too much stuff to try to measure. You can frequently use chromatography to separate it out. So, when we started, all these things were just in one dissolved phase together, one solvent, and then this sort of separated them so that we could figure out, how much of this and this, and then how does that compare to this. So yeah, the retention time is, the reason we always put that on there is, depending on what GC you're using, the retention time can be pretty indicative of what the compound is. And definitely also the order.
Also a comparison.
Yeah. So, back in, 2013, I think probably you were asking about Steve Linneman earlier. He put a paper out there in the scientific literature, peer-reviewed literature, on what we had done so far with the Hot Lake mat. And this is, as far as I know, the first paper that came out about Hot Lake since Gordon Anderson back in 1958. So it sort of put Hot Lake back on the scientific map a little bit. And what that paper primarily was doing, and unfortunately I can't answer too many questions about this.
Your name was on it.
It was, but see, I did some of the other. I didn't work on this figure, Clyde. I worked on other figures. See, but what the take-home message? One of the take-home messages from that paper was that this Hot Lake mat, when you measure it at different points over the growing season, for the most part, most of these lines are pretty stable. They're pretty flat. There's only a few organisms that really aren't present early in the season, and then they sort of come up later in the season. And maybe this guy that sort of decays at the end of the season. So the take-home message from that work was that the mat is largely the same organisms present early in the season and late in the season. We didn't think that was going to be true. Because what we anticipated, sort of why we took the samples, was we thought that, when the lake's not very salty, on a relative sense, early in the season, we thought certain organisms would grow. Then when it got really salty at the end of the season, we thought other organisms would take over. And we were way wrong on that. It's that these mats are largely composed of the same things throughout the season, which became more interesting to us. Then I can answer any questions about this one. Right? This is another paper.
Your name is first.
Yeah, see, let me come in the hot seat for answering questions. But this is some work that we did in Hot Lake actually in July 2012, I believe it was. And we're looking at the mat up there, and I'll just jump through. So what I've done, one of the fun projects I got to work on at PNNL is playing with lasers, basically, and zapping things. And that's always fun, right? This is a hair, and you see here different holes. So I ablated those holes with lasers, and I took the tiny particulates that we shot out of there when I ablated it, and I performed stable isotope analysis on it. So you all may not realize this, but you're made of a lot of carbon, and about 98.5% of the carbon in you weighs 12 atomic mass units, and that extra 1 to 1.5 weighs 13. So I spend most of my time looking at very, very subtle variations in that ratio of the different isotopes in you. And they're all completely stable. They're not decaying or anything like that. There's no health danger with those. But the different things that enzymes or people do or different reactions tend to subtly shift that ratio. So what I want to do is, this is the hair. We really get measurements on these tiny little spots in the hair. I said, okay, let's take that. It's fun to make that. Let's apply it to this Hot Lake mat. So we went up, and this is a Hot Lake mat. And the nice thing about this picture, too, I think, is that you can get a little sense of some of the different colors here. Unfortunately, I'm not very good. So you just kind of a light green, almost a whitish, a dark green, and then a lot of times at the bottom is a little bit of a pink, maybe a little bit of a purple color. And this is the top of the mat up here and the bottom down here. You can't really see it, but sometimes this white area has a lot of carbonate in it as well. And I think I know carbonate, chalk is probably the most common carbonate, but mineral phase is similar to that. Each one of these little holes is where we did some laser ablation, and I was able to do some incubations on the side of the lake, and basically we showed that inorganic carbon goes primarily into the very top of the mat. So inorganic carbon, that means photosynthesis is happening largely at the top of the mat, and okay, that's great, it makes sense, there's more sunlight hitting the top, it makes sense that there should be more photosynthesis. But then the question is, okay, these guys at the bottom that are in the shade, they're still alive, growing, and doing well. How does food they get from being fixed up here down to there? That was sort of the question that we had after, and we're trying to answer that a little bit more with some of the experiments that we're doing this year. So, are you laughing at the mat?
I said with my backyard.
Oh, this. Okay, well, we need to talk then, because we want to come to your backyard and look at this. But this is all underwater mat. This is what the mat looked like earlier this summer. I mentioned that it can produce a lot of oxygen bubbles. What happens is sometimes these oxygen bubbles get trapped in the mat, and then the mat becomes a little buoyant. Instead of being a nice, I didn't lie to you, Clyde, but instead of being a nice flat sheet on the bottom of the lake, sometimes it gets too many bubbles and it just sort of lifts off a little bit. And that's what's happening there. What we're doing now is taking mat like that, and we're not. This is something we did last year, because I didn't have time to get pictures in from this year. But we're putting them in little dishes like this and incubating them at a given temperature, and we're putting different substrates like glucose and acetic acid and things like that into these incubators. And we're trying to do measurements to figure out which one of these things go into the mat first, which ones goes in fastest, and where in the mat. So remember, the mat is, let's say, this thick. Does one substrate go in at the top and a different one go at the bottom? And then do all the substrates go in equally during the day and at night? I mentioned that during the day there's a lot of oxygen around. At night there's really not much oxygen, if any oxygen around. So how does that affect what substrates go in and where?
Is your incubation process parallel to on-site?
On-site's an interesting term. It's pretty close to on-site. There was the road to Hot Lake was closed earlier this year due to some high fire danger. So we started making plans to maybe grab the sample at Hot Lake and then perform the incubations in town. And it's nice. We have one of the other things that we're doing is we have a lot of electricity. In town we can use electricity versus at the lake we don't have any. And Hans, who's actually doing this now, he's taking the mat's this thick, and he's got these tiny little microsensors that are you got to be really close to him and squint to see them. But he's taking these tiny little microsensors and drilling through the mat, and he's measuring oxygen and the pH and things like that at the different layers in the mat in real time, sulfide, things like that. So he has all that equipment takes electricity. So we're sort of, okay, we want that. And then there's other nice things like if you have electricity, you can control the temperature of these things a little better. We have aquarium heaters and stuff like that that we use in the control.
Well, if you move your campus here, it'd be a lot easier.
Hey, I'm up for that. I like it up here. So yeah, if we could get a little satellite lab or something, this is where I'd be.
Jim, so I just want to make, I think I know the answer, but basically this is a new mat every year. I mean, you can pull these hunks out and
it grows back.
It just grows back.
It grows back, yep. And that's something that we're really interested in too, long term. We haven't really had a chance. We're thinking about how to sort of figure this out. We don't know where the mat really comes from. So there's sediment. Every year this rubbery, almost thick, gooey thing. Some people back at the lab don't touch it. They're like, oh, it's gooey and gross. Like, well, you got to see where it comes from. It's not that bad. But it's if you take it off in the fall, new mat will grow there in the spring. And we don't know if microbes are coming out of the sediment or if microbes are coming out of the water column above and colonizing on the surface. And we don't even really know which microbes get there first. And so we would like to follow up on that. But yeah, you're right. I mean, it's not...
You didn't notice those green antecedents?
No, we have not coincided with their visits yet. But yeah, we're very interested to know. But, I mean, I don't know. We've obviously we've taken chunks of mat about this big, right? And then those wounds or whatever, the mat heal very quickly. They grow back very quickly. But we don't completely know where it comes from. What's that?
It sounds like crystal. Crystal wants to be formed complete. So if you break off a piece, it forms itself back.
Reform, yeah. And I should also say that the depth of the most active mat does change a little bit throughout the year. And also, even there is very, very early in the year, there's a time where instead of being, it's hard to see here, but instead of being a little bit greener, the surface of the mat's a little bit pinker. Very early, when the light's low, early in the year, that those purple pink bacteria come up close to the surface. The other thing I should say is that this mat is a little bit greener earlier in the year, and then the sun just beating down on it all summer, it tends to be a little bit orange or even a little bit yellow by the end of the year. And we think that's just damage done to the photosynthetic pigments as that UV light streaming down over and over. So we're doing that, those incubations, and then the bad thing for me is that we're collecting samples day and night for 48 hours. So that's why I'm a little tired. So I apologize for that. But we got different people there, and it's nice being here because they're taking samples right now. As of 26 minutes ago, they were collecting the next round of samples. So this is our map. We're trying to figure out who is present in the map, what they're doing, so what they're releasing and what they're consuming, where they're doing it, so where in the thickness of that mat is the highest activity of each of those processes, when they're doing it, so again, are they more active during the day, during the night, and the answer is yes, for both. They're more active during the day and during the night for different microbes. So different parts of that microbial community change their activity level a lot depending on the day-night cycle. And then how do conditions in the mat influence what the microbes are using as their primary metabolism, and that's what Hans is doing, really, is he's taking this probe down here, he's trying to figure out what's the geochemistry in there, how's it changing, and how does that affect what reactions are favorable. So this is a little bit hard to, I made that figure so it's probably not the most clear, and I apologize for that. But this is what we did last year. The blue and red basically show, as you go further up in the scale, it shows that more of our labeled substrate was taken up into the mat. So as we're going up here, there's a lot more label in the mat than we started with at the beginning. That's great. You can see that for the most part, the amount of label goes up during the day, and then at night it goes down a little, stays level, goes down a little bit, and the next day it goes up. This is when we put inorganic carbon in. So this is what they use for photosynthesis. So it makes sense that during the day you do photosynthesis, you make fixed carbon, at night you release it, and then you put it back. The crazy thing here is the green is the number of enzymes basically that showed that same carbon being taken up. So the biomass is getting up and then up again, and although the number of enzymes that show that really takes almost a full 24 hours before you just get this almost exponential jump or increase. And we're trying to follow up with that. That's one of the things we're doing this year, trying to figure out why do we have that. Clyde's standing up, and he told me I had to be done soon, so I want to thank all the people. I don't know how many people there are at PNNL working on this project, but I think we have, depending on how probably 30, 35 people working on it. And these are some of the folks that have done a great job really helping with some of the stuff I presented today. We also have a couple collaborators at Washington State University who have been up here a few times, and we get some samples for them tomorrow. And then the other folks I really want to thank are all of you, and definitely Clyde and Sandy for always, well, every time we're in a pinch, we just go to Clyde and he's got it fixed, right? So it's really great. And also Dale Devon, the road to Hot Lake sort of cuts through his property, and he's been very nice about letting us get up there. So thank you very much, and let me know if you have any questions or anything like that. Yeah?
How deep is it?
Totally depends on the time of the year, but yeah, it's not very deep. So the deepest, earlier this year, it was probably somewhere between 6 and 7 feet deep at its deepest, and which is only a fraction of it. But yeah, so not very deep.
And there's no animal organisms.
Well, there are. It's very, very limited. There are tons of brine shrimp.
Oh.
So sea monkeys, or whatever you want to call them. Like they're just, right now they're all over. Usually they don't appear until a little bit later in the summer, like late June or July. Then there's just a ton of them. And there's still a lot of them out there right now. Some of them look like they're a little tired around a while, but yeah. And that's it. Now, every now and then, I don't know, there's weird, Hot Lake seems to be a place that some animals like to go to die. We always find like dead snakes and stuff like that. And there are like maybe deer tracks around, there's some bear tracks around.
They thought it was good water. That's
well, not to drink. I wouldn't drink it. So I don't know if that's what they're up to, but that's about it. Oh, the other thing, we do, sometimes in the spring, the top water is fresh enough that there might be a few ducks that come there for a while, but they're always gone by the time we get to summer.
Nothing preys on the brine.
No, and I think it's the salinity probably saves them. Because, you know, like fish or anything else, they can't handle it. So I think that's why the brine shrimp can actually do pretty well there.
Did you say that ammonia? There's ammonia in the lake. Is that the main nitrogen source?
No. Well, this is a conundrum sort of for us because I mean all this biomass has to have a lot of nitrogen associated with it. Every time we've made an ammonium or nitrate measurement, it's below detection levels. So now what I do know is did some experiments last summer. And we're repeating these now. Some of these organisms can fix atmospheric N2, and so we think that that's probably the primary input, but then there's only a few organisms that can do that. It's a hard process, right? And it's hard for people too, because that's why fertilizer is basically so expensive, right? It's hard to break that triple bond of nitrogen to make it into something else. But what we're really interested in is how does those few organisms that can fix it into bioavailable nitrogen, how do they then transfer it to other organisms? So ammonia might be one compound that they could use to transfer that to their neighbor, basically. Yeah.
You said rather than table salt, what two salts did you say?
It's primarily magnesium sulfate, and it's basically Epsom salts. They can, I don't know, it doesn't come from Hot Lake, but you can go to the store and buy salt that's very similar, right?
Okay, thank you, Jim. Appreciate it. Give him a hand. Oh no, I've got to have pictures. I can't. That was great. Very, very well done. Appreciate it. Thank you for coming tonight. Quite fascinating. Even if it does go over some of our heads, we know that there's people out there smarter than us figuring these things out, and we thank you for that. Let them eat. So.
Sounds like you have some stuff here to see with a lot of stuff that you're
There's a lot to learn.
So do you go back and forth between this? You spend time down at Yellowstone too? Is that the idea? The team basically just does these two sites and then stuff in the lab in between?
They send me most of the time. So yeah, they send me to get samples and bring it back.
Yeah.
Yeah. I've only been to Yellowstone a couple of times, I think, this year.
Monday morning. Baby in tow.
Shall we do it the same way? Similar picture of the one behind the mountain.
Wow.